Bismillahirrahmanirrahim. Assalamu alaikum everyone. This is Dr. Waqas Ahmed Khan, demonstrator pathology. Today we will be looking at the first lecture of genetic disorders. So let's start. So first of all, let's look at the learning objectives. At the end of the lecture, you will be able to tell what are the different genetic disorders, what is the difference between genetic and congenital disorders, what is the different classification of genetic disorders, different mechanisms and causes of genetic disorders. You will be able to discuss the chromosomal defects with special emphasis on the aneuploidy, and you will be able to define the following terminologies, which are monosomy, trisomy and polysomy. So let's start with the genetic disorders. So what is the definition of genetic disorder? Genetic disorder is a disease which is caused by a change or a mutation in the individual genes or a DNA sequence which are present on the genes. It can be manifested at birth or it can also present later in life, which means that sometimes that there is a genetic disorder at birth but it does not present itself, but it manifests itself later in the life stage of that person. Genetic disorders can be inherited from the parents to the offspring depending upon their mechanism of inheritance. So what are congenital disorders? Congenital defects, also known as the birth defects, are anomalies of the body structure, function or metabolism which are present at birth. I will put special emphasis at that these are the disorders which are present at birth. For the congenital disorder, they need to be present and manifest themselves at birth. These can be caused by two types of anomalies. Number one, these are caused by genetic factors, which can be single gene, multifactorial or chromosomal abnormalities, or they can also be caused due to the environmental factors, which are active during the embryonic or fetal development. For example, that will include maternal disease such as diabetes mellitus or thyroid or anemia or in different types of infections, and the different drugs that are taken during the pregnancy or before pregnancy. The main thing you have to consider about the congenital disorder is that they always manifest themselves at birth and can be hereditary, that is, they can be inheritable depending upon their etiologies. If they have a genetic factor, obviously, if they are sex linked or autosomal, then they can be inherited from the parents to their offsprings. But if they are due to environmental factors, then they are not inheritable. So basically the major difference between the congenital and the genetic disorder is that the congenital disorders are present at birth and they manifest themselves, and the genetic disorders, they might not manifest themselves at birth and can also show symptoms later in life. So let's look at the classification of the genetic disorders. Genetic disorders are divided into two types as shown in this table. These are the single gene disorders and these are the chromosomal disorders. So first look at the single gene disorders. These are disorders which are divided into following two types: autosomal disorders and sex-linked chromosomal disorders. Autosomal disorders, as previously studied, are divided into autosomal dominant and autosomal recessive, while the sex chromosomes are divided into sex-linked disorders, of which X-linked disorders are most common and Y-linked disorders are very rare. Talking about the second type, which is chromosomal disorders, in these types of disorders, there is a mutation in a large portion or an entire chromosomes. These are divided into two types of disorders. These can be due to the numerical differences in chromosomes, or these can be present due to mutation in the structural component of the chromosome. So basically these are divided into two types. So let's look at some examples of the autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive and Y-linked disorders. So first of all, let's look at the autosomal dominant example. These are the inherited breast cancer, inherited colon cancer, dominant autosclerosis, familial hypercholesterolemia, von Willebrand's disease, adult polycystic kidney disease, neurofibromatosis, myotonic dystrophy, Huntington disease, tuberous sclerosis. Now let's look at some examples of autosomal recessive disorders. The most common of which is cystic fibrosis. As you can see, it is very common in children. Recessive learning disabilities in children, congenital deafness, which has a multiple subtypes, phenylketonuria, spinal muscular atrophy. Now let's talk about some X-linked disorders, in which the first are the X-linked dominant disorders, in which there is example of XG blood group, vitamin D resistant rickets, hereditary motor and sensory neuropathy, incontinentia pigmenti and Rett syndrome. X-linked recessive disorders have examples of red-green color blindness, which is a common disease, non-specific X-linked learning disabilities, fragile X syndrome, Duchenne muscular dystrophy, hemophilia A, X-linked ichthyosis. As I have told you before that the white-linked disorders are very rare, so we have only one example here, which is of XY gonadal dysgenesis. So our main emphasis is on the chromosomal disorders in the genetic disorders, and as I have told you before in the previous slide that these are divided into two types. One in which there is a numerical change of chromosomes due to the addition or deletion of any chromosome number, and the other is due to the change in the change or mutation in the structure of the chromosomes. So first look at the structural chromosomal defects. These are divided into six types. First, let's look at the deletion. As the name suggests, in deletion, there is the deletion of one part or one segment of a chromosome. As you can see that this yellow color segment is deleted from this portion. The second type of chromosomal defect is translocation. In translocation, you can see both these segments of the chromosomes are interchanged with each other. This is known as translocation. The third type is inversion. In inversion, there is a break in the chromosome at two sides, which can be above and below the centromere. As you can see, this is the centromere, and it is divided into two types, pericentric and paracentric. In pericentric, there is a break on both sides of the chromosome. As you can see, one break is on this side and one break on this side of the centromere. This is the centromere, so this is known as pericentric inversion. And in paracentric, both of the breaks of the segments are on the one side of the centromere. As you can see, this is the centromere and both of these breaks are on this side. The fourth type is known as Robertsonian translocation. In Robertsonian translocation, you can see that both these short fragments of the chromosomes, this and this one, are broken from these, and the remaining two segments are fused together here, and both of the short segments are lost. There is loss of both the short segments. The fifth type is isochromal translocation, and it happens during the meiosis. During the meiosis, there is a division and there is a break between the centromere, and you can see both the short fragments are fused together and both the long fragments are fused together. This is known as isochromal chromosomal translocation. The last type of chromosomal structural defect is the ring formation, in which you can see there is fusion of both the short and both the long, which forms the ring. So our main concern is with the numerical change of chromosomes. As the name suggests, there is addition or deletion of the chromosome. These are divided into two types of defects, aneuploidy and euploidy. The most important of these is aneuploidy. In aneuploidy, there is hypoploidy or hyperploidy. Aneuploidy is defined as the addition of chromosomes or a deletion of chromosomes from the total number of chromosomes. As you know that the total number of chromosomes in the human body is 46, which is divided into 23 pairs. So the hypoploidy is divided into monosomy and nullisomy. In monosomy, there will be a deletion of one chromosome. As you can see, this is 2n-1. So one of the chromosome will be deleted from 46, having a total number of 45 chromosomes. In nullisomy, there will be a deficiency of 2 chromosomes, which is 2n-2. Then we look at the hyperploidy. In hyperploidy, it is also defined as polysomy. In trisomy and tetrasomy, both are example of polysomy. In trisomy, there will be addition of one chromosome, so the karyotype will have total of 47 chromosomes, 46 plus 1, that is 2n plus 1. And in tetrasomy, there will be addition of 2 chromosomes, as you can see here, so that will be 2n plus 2, and there will be total of 48 chromosomes. Then we have the euploidy. In euploidy, there is increase in the number of chromosomes, but in multiple of x. For example, in monoploidy, there will be x number of chromosomes. In diploidy, there will be 2x number of chromosomes. For example, if it is 2x, then it is 46 chromosomes. And in polyploidy, you can see there can be 3x, 4x, 5x. So if it is 3x, the total number of chromosomes in the human body will be 69. So as I told you before, our main objective is about the aneuploidy. So aneuploid is any chromosome number that is not euploid, which means it is an abnormal number of chromosomes in which there will be an extra chromosome, which means it will become 47, or a missing chromosome, which will become 45. Aneuploid karyotypes are given names with the suffix somy rather than ploidy used for euploid karyotes such as trisomy and monosomy. Both of them are very common. Find the terms monosomy and trisomy. As I have told you before in the previous diagram, monosomy means that there is one missing chromosome from the typical diploid cell, that is 2n-1. So there will be a total of 45 chromosomes instead of 46, as one of the chromosome will be missing. Classic example of monosomy is Turner syndrome, which has a karyotype of 45X or 45XO, where O defines a missing chromosome. So as you can see, one of the chromosome is missing. The total number is 46. So 46 minus 1, 45. Then let's come to the trisomy. Trisomy refers to the condition having one additional chromosome. It is represented at 2n plus 1, so which means that there will be total number of 47 chromosomes instead of 46, as there will be one additional chromosome. Classic example of trisomy can be seen in Down syndrome, which has a karyotype of 47XY plus 21, where plus 21 defines the chromosome number 21, which is an extra chromosome. So let's look at the definition of polysomy. Polysomy is a numerical chromosomal disorder in which there is at least one extra copy of chromosome relative to normal. For example, if the normal number of chromosome is 46, then there will be additional at least one extra copy of chromosome, which can be trisomy, or there can be more than one extra copy. There can be 2 copies, there can be 3 copies, defining as tetrasomy and so on. So what is the reason of polysomy? It is usually caused by non-disjunction, which is the failure of a pair of homologous chromosomes to separate during meiosis. But it can also be seen due to translocation mutation, which is a rearrangement of parts between non-homologous chromosomes. So as you can see, there are two main causes of polysomy. Number one is the non-disjunction, and the number two is translocation mutation. As described before, trisomy and tetrasomy both are example of polysomy, having one extra chromosome in the trisomy and 2 extra chromosomes in the tetrasomy. Common examples of polysomy are Line-Congenital syndrome and Down syndrome. So this is all about today's lecture. This is the first part of genetic disorders. In the next part of genetic disorders, we will study about some common genetic disorders such as Down syndrome, Line-Congenital syndrome, and Turner syndrome. This is all. Thank you and have a nice day. Allah Hafiz.